All right, buckle up, everyone, because today we're diving headfirst into the world of genes and neurological disorders.
We've got some fascinating research lined up.
We do. We're going to be looking at 3 different sources today.
Yeah?
A scientific paper all about ALS.
Okay.
A research report that looks at this gene called EBF3.
Interesting.
And it looks at it in both humans and fruit flies.
You're cool.
And then we're going to wrap things up by taking a look at myotonic dystrophy type 1.
Sounds good.
So a pretty wide range of stuff to cover today.
Yeah.
But one of the things that I'm really excited to dig into is how a single protein can influence a disease as devastating as ALS.
Yeah, that's a really important area of research.
It is.
ALS is a really complex disease.
Mm-hmm.
There's no cure right now, but scientists are doing some really promising work trying to figure out what's going on.
Absolutely.
At the molecular level.
So let's start with ALS.
Okay.
I think most people have heard of ALS. It's a really tough disease that affects the nerve cells that are responsible for movement.
Exactly. And one of the key things that researchers are looking at is this protein called SOD1.
Okay, so SOD1, what is it? Is this like the bad guy in our neurological story here? What makes it so important?
Well, SOD1 actually has a normal job to do in our cells.
Okay.
But when its structure gets disrupted, that's when things start to go wrong.
I see.
You can almost think of it like, a perfectly folded origami crane.
Mm-hmm.
If you bend it the wrong way, it loses its shape. It can't function properly.
Okay, I can see that.
And so that's kind of what's happening with SOD1 in ALS.
So what is causing it to misfold in people with ALS? Like, what's going on there?
So one of the major factors is this process called liquid phase separation.
Okay.
Or LLPS for short.
Okay.
And you can think of it like if you're making salad dressing.
Yeah.
And the oil separates from the vinegar.
Right.
It's kind of like that. But with proteins,
okay.
So under certain conditions, these SOD1 proteins can actually start to clump together and form these little droplets.
So instead of mixing nicely in the cell, they're basically clumping up.
Exactly.
I can see how that might not be a good thing.
Yeah. And these clumps, which are also called aggregates, are a hallmark of ALS.
I see.
And what's really interesting is that scientists are starting to understand what triggers this clumping behavior.
Okay, so spill the tea. What causes these SOD1 proteins to go rogue and form these harmful aggregates?
So there are 2 main culprits.
Okay.
Oxidation
okay.
and mutations.
Gotcha. So let's start with oxidation.
Okay. Yeah.
What is oxidation?
So you know how metal rusts when it's exposed to air and water?
Mm-hmm.
Oxidation can actually happen to proteins too.
Oh, okay.
And it can make them sticky, which leads to that aggregation.
So it's like the protein is getting damaged and it can't hold its shape anymore.
Exactly. It's kind of like rust for proteins.
Okay, that's a great analogy.
Yeah.
Okay, so what about mutations?
So mutations are actually changes in the genetic code.
Okay.
That can alter a protein structure.
Okay.
And specific mutations in the SOD1 gene have been linked to an increased risk of developing ALS.
So it's almost like having a blueprint with a mistake in it.
Right.
The protein gets built wrong from the start, and then it's more likely to clump up.
Exactly.
Okay, so it sounds like both these things, the oxidation and the mutations, they're creating these sticky proteins.
Yes.
That are primed to form those harmful aggregates.
Exactly, and that's where it gets really interesting.
Okay.
Because researchers are actually now trying to figure out ways to prevent these aggregates from forming.
Okay.
Or to break them apart once they've already formed.
I see.
It's a bit like, if you've got flour or sugar that's all clumped together.
Right.
You're trying to unclump it. It can be tricky.
Yeah.
But if scientists can figure out how to do that, it could have huge implications for treating ALS.
So understanding how these SOD1 proteins are misbehaving is crucial for developing new therapies.
Absolutely. It's all about getting to the root of the problem at that molecular level.
Really getting down to the nitty-gritty.
Yes.
All right, so that was a lot about SOD1 and ALS. But our next source takes us to the world of fruit flies.
Ooh.
And I have to admit, I'm a little curious, a little bit skeptical, perhaps, about what these tiny creatures can teach us about the human brain.
Right.
It seems a little bit far-fetched, but I'm willing to keep an open mind.
I think you might be surprised.
Okay.
Fruit flies are incredibly valuable for studying human development and disease.
Okay.
And believe it or not, we actually share a surprising amount of genetic information with them.
Really?
Yeah.
Huh, interesting.
So they're actually a really great model to work with.
Okay, so what secrets are these little flies spilling about our brains? What did they find?
So this research focuses on a gene called EBF3. which is a transcription factor, meaning it controls the activity of other genes.
Gotcha.
And it plays a really crucial role in how our brains develop. And guess what?
What?
Fruit flies have a very similar gene.
Oh, wow.
Yeah.
So by studying EBF3 in fruit flies, we can learn something about how it works in humans.
Exactly.
That's pretty cool, I have to admit.
Yeah.
But what happens when this EBF3 gene doesn't function properly? What does that look like?
So in humans, a loss of EBF3 function can actually lead to a whole range of neurodevelopmental issues.
Oh, wow.
We're talking about developmental delays, muscle stiffness, speech difficulties,
Wow.
ADHD.
Okay.
And there's even a potential link to autism.
So one gene, so many potential consequences. That's wild.
It really shows you how interconnected everything is in our bodies.
It really does.
Yeah.
But I'm still a little bit fuzzy on how the fruit flies fit into all of this. Like, what did they actually do with these flies?
So there was this group of researchers back in 1997.
Okay.
Nestor's and his team, and they used fruit flies to study EBF3, which in flies they called NOT.
NOT.
And they created these fly mutants with altered EBF3.
Okay.
Just to see how it would affect their development.
Okay. And what happened? What did they find?
And they noticed something pretty interesting.
Okay.
These flies developed specific abnormalities in their wings.
Wait, wings? I thought we were talking about brains.
I know, it might seem strange.
Yeah.
But stick with me.
Okay.
The development of wings in flies.
Okay.
And the development of the brain in humans actually share some fundamental processes.
Oh, interesting.
They both involve these intricate patterns of cell growth and differentiation.
Gotcha.
Which are guided by specific genes.
So by studying how EBF3 affects wing development in flies, we can get a better understanding of how it might be influencing brain development in humans.
Exactly.
Okay, that's a pretty clever approach, I have to say.
It is.
So what were the specific abnormalities that they noticed in the fly's wings?
So they noticed disruptions in the veins of the wings.
Yeah.
And also in the overall structure of the wings.
So it's almost like the blueprint for the wing was slightly off, and it led to these visible defects.
Exactly.
Okay, so those tiny little wing abnormalities were basically a clue that something was up with this EBF3.
Right.
What did the researchers do next? Did they kind of dig into that a little bit more?
Yeah, so they used this really cool technique called mosaic analysis.
Okay.
Where they basically created these patches of mutant cells within a normal fly wing.
So like a little patchwork quilt of cells.
Yeah, exactly.
Okay.
Where some have the normal EBF3 and some have that altered version.
Gotcha. So they're basically creating this controlled experiment within the fly's wing.
Yeah.
To compare those effects side by side.
Exactly. Okay, that's really cool.
Yeah. And by analyzing those mosaic wings, they were able to pinpoint exactly when and where
EBF3 was essential for normal wing development.
So they could see, like in certain parts of the wing, if there was a problem with EBF3, it caused a disruption.
Right.
But in other areas, it didn't seem to matter as much. So they were able to like map out the precise time and location where EBF3 was playing a critical role.
Exactly.
Wow, that's incredible.
It is.
So how does this like translate to the human brain? Like what's the takeaway message there?
So the takeaway message is that EBF3 likely plays a similar role in guiding the development of specific brain regions.
Okay.
At specific times.
Gotcha.
Just like in the fly wings. Having the right version of EBF3 in the right place at the right time is crucial.
Okay, so these tiny fly wings gave us a pretty good look into the complex world of human brain development.
Yeah, it's a great example of how studying these seemingly simple organisms can actually teach us so much about ourselves.
That's really cool. And I have to admit, you totally sold me on the fruit flies.
Good.
I'm impressed. Now, our final source for today's deep dive takes us to myotonic dystrophy type 1, or DM1.
Right.
And this condition affects not just the nervous system, but also muscles and even metabolism.
Right.
It's kind of like this prime example of how one genetic hiccup can have a domino effect throughout the whole body.
Exactly. It really highlights the interconnectedness of all of our body systems.
Yeah, it really does.
And to understand how it all works, we need to zoom in on the molecular level.
Okay.
And really look at what's going wrong in the genes.
All right, let's zoom in then.
Okay.
But before we get into the nitty-gritty, can you give us a quick overview of the main symptoms of DM1? Like, what are people experiencing?
So the hallmark symptom is muscle stiffness, which is also known as myotonia.
Okay.
And It's kind of like your muscles just have a hard time relaxing after they contract.
Oh, okay.
So imagine trying to open your hand after squeezing it really tightly.
Right.
It's slow and it's difficult.
Okay.
But DM1 doesn't just stop there. It can also lead to muscle weakness,
Okay.
heart problems,
Gotcha.
and even insulin resistance,
Oh, wow. Insulin resistance.
which you usually associate with diabetes.
Right. Okay, hold on. Muscle stiffness and insulin resistance.
Yeah,
Those seem like completely unrelated problems.
I know, right?
How can one condition cause such diverse symptoms?
So it all boils down to this single molecular culprit,
Okay.
which is an abnormal expansion
Okay.
of something called, and this is a mouthful,
Okay.
CTGN re-repeats
Okay.
within this particular gene called DMPK.
DMPK. Okay.
And this expansion is located on chromosome 19.
Gotcha.
And it messes up the gene's instructions,
Okay.
which then leads to the production of these toxic RNA molecules
Okay.
that wreak havoc on normal cellular processes.
Okay, so I'm picturing this gene that has this like extra long repeated section that's causing all these problems. But how does messed up RNA lead to all these different symptoms? From muscle problems to metabolic issues, it's like a domino effect.
It is. So think of it like this.
Okay.
This abnormal RNA acts like a molecular sponge.
A sponge. Sure.
Soaking up all these crucial proteins that are normally involved in regulating various genes.
I see.
And one of these proteins, called MBNL, plays a really key role in something called RNA splicing.
Gotcha.
Which is a process that makes sure the correct instructions are sent for building proteins.
So the toxic RNA is hijacking these essential proteins, preventing them from doing their jobs?
Yes.
Okay, and this disruption in RNA processing, that's what's causing the domino effect.
It is.
Okay, so explain that to me then.
So in muscle cells, this disruption leads to the missplicing of RNA, which then contributes to that myotonia symptom.
That difficulty relaxing the muscles.
Right. But here's where it gets really interesting. This disrupted RNA processing can also affect the production of insulin receptors, which are crucial for regulating our blood sugar levels.
Okay, so that's the connection between the muscle stiffness and the insulin resistance.
Exactly.
It all comes back to that messed up RNA.
Yes.
It's amazing how one tiny molecule can have such a huge impact on so many different systems.
It really shows you the interconnectedness of our bodies.
It does.
At that molecular level.
Yeah, it really does.
And, understanding those underlying mechanisms is what's driving the research for potential treatments.
That makes sense. So speaking of treatments, what does the research say about potential therapies for DM1? Is there hope on the horizon?
There is, yeah.
Okay, good.
So there are three main categories of treatment approaches.
Okay.
First, we have theoretical treatments.
Okay.
Like gene editing or silencing.
Okay.
Those hold great promise.
Yeah.
But they're still in those early stages of development.
Okay, so gene editing would be like actually correcting that faulty DMPK gene. And gene silencing would be preventing that toxic RNA from being produced in the first place.
Exactly.
Okay, those sound like really promising approaches.
They do.
But it sounds like they're still a ways off.
Yeah.
So what about more immediate options? What's out there?
So then we have candidate treatments.
Okay.
Such as RNA blockers or RNA interference.
Gotcha.
And these are showing a lot of promise in animal models.
Okay.
But they haven't made it to human trials yet.
Okay, so explain those a little bit more. Like, how do those work?
So basically, RNA blockers work by binding to that toxic RNA and preventing it from interfering with other molecules.
I see.
And then RNA interference aims to destroy the toxic RNA altogether.
Okay, so some interesting possibilities in the pipeline there.
Yeah.
But what about practical treatments? Like, what's available right now for people that are dealing with DM1 today? What can help them?
So there are some existing drugs that are being repurposed for DM1.
Okay.
And they're showing some good efficacy in managing those symptoms.
Okay, that's good.
Yeah, so for example, mexiletime, which is a drug that's typically used to treat irregular heartbeats, has been found to actually help alleviate muscle stiffness.
Oh, wow.
In some people with DM1.
So while there might not be a cure yet, there are options available to help people manage their symptoms.
Yes.
And researchers are actively working on developing new and more effective therapies.
Absolutely.
That's really encouraging to hear. It's encouraging to hear that there's active research going on and even some treatments out there for DM1. It seems like the more we learn about these complex disorders, the closer we get to finding effective ways to really address them.
Yeah, definitely. And you know what's interesting is that all three of the sources we've looked at so far, even though they focus on different neurological conditions, they all highlight this common theme, how important it is to understand those molecular mechanisms that are at play.
That's true, yeah. We saw how that one misbehaving protein, that SOD1, can contribute to ALS.
Right.
And then we saw how studying EBF3 in fruit flies can give us insights into human brain development.
Exactly.
And then finally, we learned how messed up RNA can have that domino effect throughout the body, leading to a range of symptoms in DM1.
Absolutely.
So we've kind of zoomed in, right?
Yeah.
From those symptoms and diagnoses down to the world of molecules and genes.
It's pretty amazing.
It is.
And, you know, this molecular understanding.
Yeah.
That's what's driving progress in developing new therapies.
Right.
And hopefully cures down the line.
That's the hope.
Yeah.
It's pretty mind-boggling to think that these tiny molecules and genetic sequences hold the keys to understanding and treating such complex diseases.
It is. It's really remarkable.
It is.
Think about it, you know, it really highlights the power of scientific inquiry, right?
It does.
That by digging into the tiniest details of biology, we can uncover these profound insights into human health and disease.
It really makes you wonder what other surprising connections and breakthroughs are waiting to be discovered.
Oh, absolutely.
If we can learn this much from a fruit fly, right?
I know, right.
Imagine what we might learn from exploring, like, the rest of the natural world.
Exactly. There's a whole universe of knowledge out there just waiting to be uncovered.
There is.
And with, you know, the advancements in technology and research, we're in a better position than ever to explore it.
That's true. That's a very good point.
Yeah.
Well, that's a very inspiring thought to end on.
He is.
I think.
Yeah.
So today we delved into the world of neurological disorders, exploring those intricate connections between genes, molecules, and symptoms.
Yeah.
We saw how understanding those connections is really crucial for developing new treatments and finding cures for these debilitating conditions.
Exactly.
It's a good reminder that even with these complex challenges, scientific progress offers hope.
Absolutely.
And that promise of a brighter future for people who are affected by these disorders.
I agree.
And on that note, we encourage you to keep exploring these fascinating topics and to really marvel at the wonders of science.
Yes.
The journey of discovery never ends.
Never.
And there's always something new to learn.
For sure.
So until next time, happy deep diving.
Happy deep diving.
Yeah. It's really good to hear that.
Yeah.
That there's that active research happening and there are treatments, you know, being worked on and being used.
Yeah, definitely.
But I think it's really interesting to point out that, you know, we've looked at these three sources today and, you know, on the surface, they're all about these different neurological conditions, but they're all highlighting the importance of understanding those molecular mechanisms.
Absolutely. That's so key.
That are at play, right?
It is.
So we saw how that one misbehaving protein, SOD1, can contribute to developing ALS. And then we saw how studying EBF3 in fruit flies can actually give us insights into human brain development.
Exactly.
And then finally, we learned how messed up RNA can have that domino effect throughout the body, leading to all those different symptoms in DM1.
Absolutely, yeah. It's fascinating how it all ties together.
It is. It's kind of like we zoomed in from the level of, you know, symptoms and diagnoses down to this world of molecules and genes.
Exactly, like down to the nitty-gritty.
And this molecular understanding is driving progress in developing those new therapies and cures.
Absolutely. That's where the breakthroughs are going to come from.
Yeah, it is. It's just amazing to me.
It is pretty remarkable.
To think that these tiny little molecules and genetic sequences hold the keys to understanding and treating these really complex diseses.
It is. It really highlights the power of scientific inquiry. You know, just digging deep into the tiny little details of biology can help us uncover these huge insights into human health and disease.
Absolutely. And it makes you wonder, what other surprising connections and breakthroughs are just out there waiting to be discovered.
Oh, absolutely. I mean,
if we can learn this much from studying a fruit fly.
Right. It's incredible.
Imagine what we can learn from,
Yeah.
exploring the rest of the natural world.
I know. There's a whole universe of knowledge out there.
There is.
Just waiting to be explored.
Just waiting to be uncovered.
Yeah.
Yeah. And, with the advancements in technology, in research, I mean, we really are in a better position than ever to explore that.
We are. It's a really exciting time to be in science.
It is. It is.
For sure.
Well, that's a very inspiring thought to end on.
Yeah, it is.
So today, we delved deep into the world of neurological disorders.
We did.
We explored the connections between genes, molecules, and symptoms.
Absolutely.
And we saw just how important understanding those connections are for developing new treatments and ultimately for finding those cures.
Exactly.
For these debilitating conditions.
Yeah, and I think it's a good reminder that even in the face of really complex challenges, scientific progress is continuing.
Yeah.
And it offers hope for the future.
It does.
For people who are affected by these disorders.
And on that note, we encourage you to keep exploring these topics.
Yes, keep learning. Keep diving deep.
Keep marveling at the wonders of science, right?
Absolutely.
The journey of discovery never ends.
It doesn't.
And there's always something new to learn.
For sure.
So, until next time.
Yeah.
Happy deep diving, everyone.
Happy deep diving.